All right, we're on our last page of notes to wrap up the membrane section, and this is how the tonicity, which is your hypertonic, hypotonic, and isotonic solutions of the solution, will affect the cells. Cells are affected by their hypertonic, hypotonic solutions, and organisms must really regulate the amount of solute and solvent concentration. Another way to look at this, and kind of a nice little trick, is if you actually put something into a hypotonic solution, you're going to make that cell swell. So that's why you see a capital O, making that cell get bigger. If you place that cell in a hypertonic solution, you're going to make the cell shrink. Because remember, that water in the cell is going to want to go to the hyper party. So it's going to leave that cell and make it really small, that little e. So you have some examples pictured below. In the first picture we see, if we place something in a hypotonic solution, so that's out here, our hypo. We have a cell. There's lots of stuff going on in the cell. We have lots of different protein molecules, sugar molecules, all of that. So we have a lot going on. So this would be our hypertonic solution. And you can see our water is wanting to go to that hyper party. And so eventually that cell can't handle it and it will lyse. So this is our animal cell example. Now your plant cell is a little bit different because of the cell wall. So here we see your plant cell. If we place it in a hypotonic solution, the water still goes in, but now the plant cell is protected by the cell wall and will actually expand and push against the cell wall, giving it a nice, what we call, Turgor pressure. Or you'll see it is turgid, or normal for a plant cell. You want it to be a little expanded, make those leaves look good. In your isotonic solution, we'll have some movement going in, some movement going out, but we're keeping it pretty equal, and that also looks the same in your plant cell. If you place these cells in a hypertonic solution, where we have a lot going on the outside, lots of solutes swimming around, and it's more than what's happening in the cell, then the water is going to want to leave to go to the hyper party. So we say it gets smaller or shrivels up. In plant cells, we see a similar thing happen, except we have a nice fancy word for it. We say it is plasmolysed, which means all of that water has left, gone to the hyper party, and now we have a really small shrink cell. So we have a few definitions that you need to know. Different solutions lead to cytolysis. Cyto means cell. Lysis means to break. So it literally means breaking apart the cell or destroying it. Or crenation or plasmolysis. So animal cells that burst will lead to, of course, death. This is called cytolysis. Animal cells that shrink up and eventually die is called crenation. Think of the word cremation. Actually taking the body, turning it into ashes, making it smaller. So that's easy for us to remember. And then a plant cell shrinking, P for plant. This is plasmolysis. And kind of a side note here, your plant cells can experience what we call Turgor pressure, actually pushing against the cell wall. So here we see an example of that in the first picture. Turgor pressure is a pressure exerted on the cell wall by water. If you have high turgor pressure, you have tight, crisp plant cells. So think really good lettuce or something, that's what it reminds me of. If you have low turgor pressure, think of like really old, kind of wilted, nasty plants. Something that you wouldn't want to eat in your salad. So just remember, plant cells require more of a hypotonic solution to help stay full and tight, keeping that plant upright. Whereas animal cells are a little bit different. They don't have that protection, so they need to be more in an isotonic environment. Otherwise, we're going to see that cytolysis or that crenation, depending on what you put them in. And that concludes the last page of notes.